Hi everybody. Thank you for the opportunity that give us to chance to talk about what's the latest progress of the European Nucleotide Archive, which we frequently call ENA, and also give us a chance to talk about the future plan for you, future plan. We need your input to address the sustainability together. And okay, do I? Okay, click up here. Okay. Um, you may or may not know that ENA is one of the three founding members of International Nucleotide Sequence Database Collaboration. There are three founding institutions: NCBI in Bank, DDBJ in Japan, as well as us in Europe. We are global comprehensive scientific record of the sequence and related data, and also over the years that we started applying and successfully make the platform to specific scientific domains. As any of the archives that we have submission side as well as a distribution side, and the genome data archived in ENA or ISDC databases are roughly in three categories: read, assemblies, and annotations. For this data to be accessible, findable, reusable, interpretable, we build comprehensive metadata models, which I'm going to elaborate a little later. We established the existence since early, I think since late 1980s. We always make sure our data is fair, which is findable. We have the ENA metadata model. I'm going to spend a minute here to talk about. We make sure we not only enable the third-party access, we make the third-party access as freely as easily as possible. And we connect ourselves with other data sources to make sure the data is interpretable and enable the downstream analysis extensively. I'm going to give two examples near the end. The community members entrust us as the long-term data archival, and also the publishers frequently require the genomic data to be submitted to ISDC databases or to ENA before they get published. By the way, that three founding members in ISDC, we interchange data together. So the data is submitted to one member and will be populated into two other members. On the right-hand side, this is a metadata model. All the research is captured by a study, and if you have the raw sequences, you can submit to us as a runs, and the description of your raw sequences can be captured as the experiments. And then your methods to prepare the sequence can be captured as samples. And if you do your own in-depth analysis, of course, the analysis results can be submitted as analysis. And the arrows there is depicting the dependencies of each of these model objects. And as you can see that everything is pointing back to study. So this way we make sure that all your data submitted separately, all well connected, and can be depicted and can be found through studies. Here's a little bit of elaboration of the three major categories of the data in ENA or in ISDC databases. In the reads, you can have the runs, which is the raw sequences, as well as the experiments to describe the raw sequence methods. And of course, for assemblies, that you put the sequence and reads together into context, chromosomes, as well as the genomes. And your in-depth analysis in the annotation category, annotate coding sequences, non-coding sequences, etc. And the dark blue boxes down there is the metadata model that are helping you to allocate, identify, and for the data, the metadata model I just talked about in previous slide. The keynote speech yesterday afternoon that Janet was kindly acknowledged that ENA has been used to help their studies. They submitted Neanderthal genomes as well as ancient human DNA into ENA. I was pretty happy to hear that ENA is being connected with such groundbreaking research into the human history. This example, it's one of my favorite examples, is also related to the evolution. This model is a 3D model of a virus, the polyoma virus, which means that obviously means that many tumors. Some research groups that has discovered new viruses in fish, cow, and sheep, etc. Obviously, these are all the vertebrates. And then they also did the sequence search against the ISMB database and identified more new species which people was not aware of, but submitted a sequence anyways. In those sequences, the genomes for 7 invertebrate hosts. This proves a pretty strong evidence that the polyoma virus existed in the last common ancestor of the arthropod and the vertebrate. So old data can play new tricks and can give the new insights of enable new scientific discoveries. Research communities entrust us with all their data so that ENA has been growing rapidly and has a very big size. I'm not going to read all the numbers there, and it's been recognized as a core biodata resources by Elixir and the new foundation of the global core data core biodata resources. In the recent years, or actually in a very long time, as you see in the graphs there, that the ENA has been growing consistently for 30 to 40% year over year, and which could exceed 150 petabytes within 5 years on this projection, which is a good news, and to a certain extent it scares us as well. For the very simple reason that just to store this much of data, it could cost us over $37 million a year by 2027. Obviously, we need to make sure that ENA is growing as to support the community, and also we need to make sure that the ENA is sustainable. We want to make sure that ENA is running efficiently. So we set up 3 goals ourselves. The first 2 goals are more for our internal goals. One is to increase operational performance, obviously, and then also on the dev side, we want to improve and simplify the system to make sure it's sustainable. So dev and ops. And the third goal, I want to emphasize that we want to tailor the services to fit the modern sequence analysis workflows. As you're aware that the ENA has been started and evolved since the 80s. So there's a lot of historical implementations, etc. So, and some of the things were designed to work for the more of, I should say, ancient analysis, and it's still there, and we want to evolve that to work better with the community as of today. So to tailor the service to fit the modern workflow, we studied analysis ourselves on the SARS-CoV-2 pipelines. You may or may not be aware that we have been analyzing the SARS-CoV-2 genomes submitted to us in the past 2-3 years, and we were able to analyze millions of genomes and make the analysis results immediately available back to the community publicly, openly. Here are the two pipelines that for this workflow. One is to design for the Illumina platform. The other one is designed for the Nanopore platform. Both of them retrieve the raw reads submitted by the community. There are over 150 institutions and I think it's over 150 countries. Anyways, my brain is a little fuzzy here. Hundreds over in, about 200 community institutions in 150 countries that are submitting the raw sequence to us. We take those sequences and perform the analysis and to generate assemblies and annotations and submit back to ENA, and they're all captured by the studies, so people can look at the raw sequence and relate them to the assemblies as well as the annotation results in those studies. These pipelines following the same pattern that everybody else is doing, that we intentionally designed the workflows just as if that we are not part of ENA. So to get the data, you don't need account or login, and we have the ENA browser. You can view and explore the data, and as well as the search and retrieve this smaller set of the data in the web UIs. And then the metadata and large amount of data you can download via the REST APIs and or some more generic downloading tools. And we also created an ENA file downloader to make this process a little easier for the community. If you're trying to submit the sequences to us, obviously, you need to identify yourself and claim your ownership, etc. So you create a submission account and create a study accession, so you can identify all the rest of the sequences, analysis, etc. belongs to this study. So we have the submission services, which you can do it in graphic user interface. We call webbing interface. And you get, once you submit it, after we done initial analysis and process, and we'll provide you the submission report to detail what you have submitted and if and when and how we archive your results. And your results can be accredited through the ORCID IDs. If you provide us with ORCID ID, and then your ownership and the discovery can be identified that way, just like as if the publication papers. For the SARS-CoV-2 analysis, we also have created, in the past 2 years, we also have created the analysis submitter to generate the analysis XMLs and the submission XMLs automatically to make the life a little easier. And also over time that we are simplifying the process for help you generate the metadata to identify your data. Yeah, this is the diagram that is showing that how what we have done in the past 2-3 years, millions of genomes being analyzed this way, and we also that was the single taxonomy ID. So we also started analyzing the metagenomics, which is a slightly different workflow for genome analysis. And it's going similar approach, getting the raw reads and analyze the context and bring the assemblies together, and also if the assemblies can be identified and can be submitted as metagenomic assembled genomes back to ENA. Again, they're all grouped and the studies to be retrievable later on and make the access a little easier. This shows that how Magnify platform as well as the community is submitting their raw sequences as well as the analysis results to ENA. And we further expanded the analysis to see how ENA is connecting and interacting with the other data sources within EBI. As you can see that it's a pretty complicated connections between all these different data sources. What the red represents those data sources reading from ENA, and those blues that represents those data sources submitting the data back to ENA, and users can find the information from ENA to these data sources, as well as from these data sources connect to ENA. We have a survey which if you are interested, and we strongly recommend you to answer our survey to help us understand how you are using ENA in the modern context and to help us to tailor the services to serve you better. Thank you for your attention. I should